What's going on? This is Braylon. Today I'll be showing you the sliding filament model of contraction. And to put this into more detail, the sarcomeres. So, for the basic structure, for purple, I have the actin, the purple serving as the actin. The red ties are serving as the myosin, and the green ties are kind of just serving as like a section of a sarcomere, and the blue ties serve as the M line, which goes directly down the middle of the A band. And for the bands, the A band spans from one end of a myosin filament to the other. So here we would have our A band, and our I band would be from the end of one of our myosin to the end of another myosin in a sarcomere. So when a muscle contracts, the actin is pulled along myosin toward the center of the sarcomere until the actin and myosin filaments are completely overlapped. So they never touch. None of these fibers ever touch. They overlap. So when our muscles contract, none of our actin will never come into contact with our myosin will never come in contact with our actin, and so and so. Going on from that, when a sarcomere shortens, some regions shorten whereas others stay the same length. A sarcomere is defined as the distance between two consecutive Z discs or Z lines, and that would just be to either side of this. We would have our Z line, which separates this sarcomere from another sarcomere, creating our I band. So when a muscle contracts, the distance between the Z discs is reduced. The H zone, the central region of the A zone, contains only thick filaments, myosin, and is shortened during contraction. The H zone becomes smaller and smaller due to the increasing overlap of actin and myosin filaments, and the muscle shortens. And what this means is when the muscle is fully contracted, the H zone is no longer visible. So to represent this, when we contract our muscles, our actin filaments are going to slide in past one another, never overlapping. They never merge or touch one another. They just slide past one another, hence why it's called the sliding filament theory. So slide those over, slide that over. These all come in, and yeah, that is the sliding filament theory represented in a sarcomere. What's up? It's Braylon again, and this is just a follow-up to the first video that I did, in which I showed sarcomere contraction and what happens with the bands and sarcomeres and the myosin and actin. And this is going to be a video on the ions and chemicals that get released in muscle contraction. So here we have a actin filament, and for our actin we are going to be using the green tie, and for our tropomyosin we're going to be using the red tie. Tropomyosin covers actin, and specifically it covers the myosin binding sites in which our troponin complex will bind to in order to allow muscle contraction. So to give more details on what troponin and tropomyosin do, they are two proteins which regulate sarcomere contraction via calcium binding. When calcium ions are present, calcium binds with troponin and removes tropomyosin. It exposes the myosin binding site in actin. When the muscle is relaxed, tropomyosin blocks myosin binding sites in the actin filaments. So, to take you through the process of muscle contraction, firstly, a nerve impulse travels down a motor neuron axon. And then the motor neuron terminal releases the neurotransmitter acetylcholine, or ACH. ACH binds to ACH receptors, and after that, the sarcolemma is stimulated and a muscle impulse travels over the surface of the muscle fiber and deep into the fiber through the transverse tubules. This muscle impulse reaches the sarcoplasmic reticulum and calcium channels open. This is when our calcium gets released and will look for troponin to bind to. Calcium ions, and for this I have not pink, I have green, a little green ball to represent the calcium. And calcium ions diffuse through the sarcoplasmic reticulum into the sarcoplasm and bind to troponin molecules, causing it to pull tropomyosin. So what this does is tropomyosin is already going to be alongside our actin filament, and once calcium is released, calcium is going to bind to our troponin, and this allows for our troponin to bind to our tropomyosin, which results in the tropomyosin freeing or allowing or opening up the myosin binding sites. So following the binding of calcium and the removing of tropomyosin, this leads to energized myosin heads attaching to an actin myofilament, forming a cross bridge. And after this, ADP and phosphate are released, and the myosin head pivots and bends. As a result, it pulls the actin filament toward the M line, which we know from the previous video, the M line is the center of a myosin. And after ATP attaches to myosin, the link between myosin and actin weakens and the myosin head detaches. Following that, ATP attaches to myosin, the link between myosin and actin as ATP is hydrolyzed to ADP and phosphate, the myosin head returns to its pre-stroke high energy. And after our muscles have contracted and we're beginning muscle relaxation, calcium ions begin to actively transport back into the sarcoplasmic reticulum, so our calcium ion will go away and detach from our troponin. Our ATP breaks linkages between the actin and myosin filaments without breakdown of the ATP itself, as to not waste any energy. Following ATP breaking the linkages, the actual breakdown of ATP occurs and begins to unform the cross bridges. Following that, troponin and tropomyosin molecules inhibit the interaction between myosin and actin filaments. So what once during muscle contraction, when our actin and our tropomyosin were separated, now the tropomyosin goes back to covering the myosin binding sites. And after that, the muscle fiber remains relaxed, so everything goes back to the way it was before muscle contraction began, and our muscle fiber stays relaxed.